The second part I will discuss here is the protease inhibitors. On the top of this slide, we can see the sequence of the typical protease. There are two typical proteases can be targeted as the therapeutic targets for COVID-19. One is called papain-like protease. It's abbreviated as PL-PRO. Another is main protease. We can either call it M-PRO or 3CL-PRO. The left side is the structure of the SARS-CoV PL-PRO in complex with an inhibitor GLR0617. It shares 86% amino acid homology with the novel coronavirus. On the right part shows the dimer structure of SARS coronavirus M protease. It has even a higher homology, shares 96% amino acid with the novel coronavirus. This high similarity or homology means we can use those known structure to help us design new inhibitor against novel coronavirus. On this slide shows some structure of M-PRO inhibitors. For example, here we can see 11A, 11B. Here is the structure of 13B, 13A. And here is the in vitro enzymatic inhibitory effect of 11A, 11B. On the top of this slide, we see clearly the structure of 11A and 11B. It have quite good inhibitory effect against M protease. And for compound 13B, it can inhibit the purified recombinant novel coronavirus M-PRO with an IC50 value around 0.7 micromolar. And here on the lower part is a table of preliminary PK data has been conducted and shown here. Besides those drugs, other potential drugs have been proposed for treating COVID-19. On this slide, we can see disulfiram, lopinavir, Camostat. First, let's look at the disulfiram. The structure is relatively simple, and on the right part, we can see the binding mode of disulfiram to SARS coronavirus PL protease. We see here, it can form a covalent adductor with the PL protease. It will form a diacyl dicyl carbamate moiety on the active site of the cysteine 1 turf here. Then it can prevent downstream acylation and thereby inactivate the enzyme. This drug can inhibit DOB activity of PL protease. For SARS coronavirus, it works as a competitive inhibitor. In contrast, for MERS coronavirus, it can work as a non-competitive inhibitor. This slide shows the structure of two other protease inhibitors. One is lopinavir, the other is ritonavir. these two compounds sometimes can be used as a combo. The first drug, lopinavir, provides hydrophobic side chain which can fill 4 pockets of the active site that are closest to the catalytic ASP residue. And it was believed that those two compounds can inhibit PL protease of coronavirus. Actually, it has been tested in the clinical trial. This table shows the combo use of these two compounds can reduce greatly viral loads and improve the clinical symptoms for patients with relatively mild symptoms. However, for the patient with serious symptoms, the treatments cannot provide significant improvement. Except those compounds, there's a research group published a preprint paper. It described using molecular docking-based virtual screening to screen based on the structure of the novel coronavirus M-Pro and PL-Pro structure. The library consists of more than 11,000 compounds. Those compounds are either FDA approved or has entered into clinical trials. On this table, the research has reported quite a lot potential drugs can work as potential inhibitors for M-Pro or PL-Pro. However, further evidence are needed to support these drugs' use in clinical.